Welcome to Lecture 12.2, in which we will provide an overview of epigenetics and regulation of gene expression. Our learning objectives for this video are to distinguish between general and specific transcription factors, understand how chromatin structure affects transcriptional activity, define epigenetics and understand how different epigenetic modifications affect gene expression, understand how mRNAs can be regulated by small RNAs, and understand how proteins can be regulated by phosphorylation and ubiquitination. Unlike prokaryotes, eukaryotes are compartmentalized, with transcription occurring in the nucleus and translation occurring in the cytoplasm. The DNA is also organized differently, and these allow for more precise regulation of gene expression. Eukaryotes generally also have many different types of cells specialized for different functions. This image shows the 4 basic tissue types in humans, and the types of cells in each category. Each cell needs to know which genes they need to express and when. For example, you wouldn't want bones growing inside your intestines. This makes regulation of gene expression in eukaryotes critically important for everyday life. Like prokaryotes, eukaryotes can be regulated at the transcriptional level by DNA binding proteins. Recall that various transcription factors called TF2 bind to the promoter and recruit RNA polymerase II. These are general transcription factors that are needed every time transcription occurs. However, there are also specific transcription factors that will bind to DNA only at specific times or in specific tissues. They can act as activators to increase transcription or as repressors to decrease transcription by blocking other transcription factors. Like general transcription factors, specific transcription factors can bind to promoters next to the gene they regulate or regions close to the promoters. They can also bind to enhancers, which are DNA sequences that are named for enhancing transcription of a gene, but are linearly far away from the gene they regulate. Something to keep in mind is that DNA is organized in 3D. So in 3D, the DNA can loop so that the enhancer and the transcription factor binding it is spatially close to the gene being regulated. The structure of DNA in chromatin also affects whether or not it is transcribed. Recall that DNA in the cell is found wrapped around histone proteins that form a complex called the nucleosome. They are then further folded and condensed into chromatin. Heterochromatin refers to chromatin that is highly condensed, such as during cell division. When the DNA is this tightly packed, it is considered to be closed off and not available for transcription. Chromatin that is less condensed is referred to as euchromatin. Euchromatin is generally considered to be open chromatin and is associated with more transcription. Epigenetics refers to how genes are modified without changing the DNA sequence. Epi means on top of, and refers to how these modifications are not made to the bases, but can be made on top of the DNA, or the proteins the DNA is wrapped around. These modifications then affect chromatin structure and transcriptional activity. While the epigenetic modifications can be passed on as a cell divides, unlike mutations, they are reversible. Epigenetics is often thought of as a mediator between our genes and the environment. Environmental factors can lead to changes in epigenetic markers, which can then be passed down to future generations. Because of this, there is a strong link between epigenetics, health, and disease. One type of epigenetic modification is DNA methylation. This usually involves the addition of a methyl group to a cytosine, although other nucleotides can be methylated as well. While this modifies the cytosine, it does not affect its ability to pair up with guanine, and therefore does not affect the DNA sequence itself. DNA methylation on the promoter is generally associated with more condensed chromatin and repression of gene expression. Another type of epigenetic modification is the covalent bonding of various molecules to the histones that DNA is wrapped around. There are many possible types of modifications, but the most common are acetylation and methylation of lysines. Lysine is a positively charged amino acid, so the negatively charged DNA is attracted to the lysine and wraps more tightly around the histone, closing it off and preventing transcription. The addition of an acetyl group neutralizes the positive charge on lysine so that the DNA is more loosely wrapped and available for transcription. This is why histone acetylation is generally associated with increased transcriptional activity. Histone acetyltransferases, abbreviated HATs, are responsible for adding acetyl groups onto a histone, while histone deacetylases, abbreviated HDAC, are responsible for removing acetyl groups. Methylation of histones can increase or decrease transcriptional activity, depending on where the methylation is and how many methyls are present. Up to 3 methyl groups can be added onto an amino acid in the histone. Methyltransferases are responsible for adding methylation, while demethylases remove methylation. Previously, we discussed how when there are 2 X chromosomes, one of the X chromosomes becomes inactivated. This is actually also a form of epigenetics. X inactivation requires a gene called EXIST, which stands for X inactivation specific transcript. This gene actually produces a long non-coding RNA, which is a transcript like mRNA, but does not code for a protein, hence the non-coding. The EXIST long non-coding RNA coats the X chromosome that gets inactivated, recruiting proteins that help modify the histones such that the chromatin becomes incredibly compact and unavailable for transcription. Although the synthesis of mRNA from a gene indicates that it is transcriptionally active, the mRNA does not always get translated into protein. Small RNAs are short RNA sequences that bind to mRNA to negatively regulate their expression. One type of small RNA is microRNAs, which often bind to the 3' end of mRNA. They can induce destabilization and degradation of mRNA by slicing it in half or by shortening the poly A tail, and can also inhibit translation of the mRNA. While these microRNAs are mostly complementary to their mRNA target, they often do not perfectly base pair with the mRNA. This allows a single microRNA to regulate multiple mRNAs. Small interfering RNAs, generally called siRNAs, are similar to microRNAs, but generally have perfect complementarity to their targets. They are also generally used to target the genes that they are derived from. Proteins can be modified in many different ways. One way that proteins are commonly regulated is through phosphorylation by kinases. Phosphorylation can make a protein more or less active, depending on where on the protein the phosphorylation occurs. For example, recall that p53 is a tumor suppressor that helps ensure that cells with damaged DNA do not replicate. Usually, p53 is kept inactive by another protein called MDM2. When DNA damage occurs, signals are passed through the cell, and some of these signals tell kinases to phosphorylate p53. This phosphorylation causes the MDM2 to let go of p53. Now p53 is active and can act as a transcription factor that promotes transcription of genes involved in DNA repair. Another common modification is ubiquitination. This is the addition of a ubiquitin group onto an amino acid on the protein. The ubiquitin acts as a mark that targets the protein to the proteasome, which is like a machine that shreds proteins into amino acids. MDM2 is actually a ubiquitin ligase, which is an enzyme that sticks ubiquitins onto proteins. When there is no DNA damage, MDM2 will add more ubiquitins onto p53, sending it to the proteasome. In this video, we learned that eukaryotes have general transcription factors for all transcription and specific transcription factors that can bind to the promoter or enhancer to regulate transcription. Heterochromatin is closed and condensed chromatin that is not transcriptionally active, while euchromatin is open and less condensed chromatin that is more transcriptionally active. DNA methylation is an epigenetic mark that is generally associated with decreased transcriptional activity, and histones can be modified to further modulate gene expression. Small RNAs such as microRNAs and siRNAs can target and degrade mRNAs so that the transcript does not get translated. We learned that proteins can be activated or inactivated by phosphorylation and degraded by the proteasome after ubiquitination. Regulation of gene expression is incredibly important, and there are of course many more ways genes and proteins can be regulated. My hope is that what you've learned in this lecture provides some additional appreciation to all the controls that are in place to make sure our cells are expressing the proper proteins and functioning correctly. Thanks for watching.